Hello friends and welcome. In today's program, we will talk on the topic mechanism of toxicity. The main objectives of today's program will be number first to understand what is toxicity and toxicant, how a toxicant enters an organism, number third, how it interacts with target molecules, number four, what are the effects of toxicant, number fifth, what are the chemical factors that cause cellular dysfunction, and number six, what is necrosis, and then what is apoptosis, and then we will know how ROS, RNS cause dysfunction, and in the last, mechanism of repair or disrepair of a cell. So let us begin with the introduction. In a general sense, the toxicity of a substance could be defined as the capacity to cause injury to a living organism. A highly toxic substance will damage an organism if administered in a very small amount. A substance of a low toxicity will not produce an effect unless the amount is very large. Thus, toxicity cannot be defined without reference to the quantity of substance administered or absorbed, or what we call as dose. The way in which this quantity is administered, for example, inhalation, ingestion and distributed in the time, for example, single dose, repeated doses, the type and severity of injury and the time needed to produce that injury. A toxicant is any chemical that can injure or kill humans, animals or plants. A poison. The term toxicant is used when talking about the toxic substances that are produced by or are a byproduct of human-made activities. An understanding of the mechanism of toxicity is of both practical and theoretical importance. Such information provides a rational basis for interpreting descriptive toxicity data, estimating the probability that a chemical will cause harmful effects, establishing procedures to prevent or antagonize the toxic effects, designing drugs and industrial chemicals that are less hazardous, and developing pesticides that are more selectively toxic for their target organisms. So, let's focus now on physiochemical properties of toxicants. In 1854, the Russian toxicologist A.V. Pelikan started studies on the relation between chemical structure of a substance and its biological activity. The structural activity relationship, what we call as SAR. The chemical structure directly determines physiochemical properties, some of which are responsible for biological activities. And when we talk of physiochemical properties, generally we talk of melting point, boiling point, vapor pressure, dissociation constant, Nernst partition coefficient, activation energy, heat of reaction, reduction potential, etc. And we also talk of electrical ionization potential, dielectric constant, dipole moment, mass charge ratio, etc. And when we talk of structure, the number of bonds, number of rings in polycyclic compounds, extent of branching, etc. For each toxicant, it is necessary to select a set of descriptors related to a particular mechanism of activity. Friends, let's focus now on mechanism of toxicity. Toxicity involves toxicant delivery to its target or targets and interactions with the endogenous target molecules that may trigger perturbations in the cell function and or structure, or that may innate repair mechanisms at the molecular, cellular or tissue levels. As a result of the huge number of potential toxicants and the multitude of biological structures and processes that can be impaired, there are a tremendous number of possible pathways that may lead to toxicity. The most complex path to toxicity involves more steps. And first step is delivery from the site of exposure to the target. Second step involves reaction of the ultimate toxicant with the target molecule. Third is cellular dysfunction and resultant toxicity. And fourth is repair of disappear. When we talk of step first, that is delivery from the site of exposure to the target, then we talk of deposition of the toxicants, and which involves absorption, distribution, excretion and toxication. And when I talk of absorption, apart from the local effects at the site of contact, a toxicant can cause injury only after it is absorbed by the organism. Absorption can occur through the skin, gastrointestinal tract, the lungs, and several minor routes. Furthermore, the nature and intensity of the effects of a chemical on an organism depends on its concentration in the target organisms. And in absorption, we have tissue diffusion, filtration, spatial transport, and endocytosis. Absorption is the uptake of a substance from the environment into the organism. The term usually includes not only the entrance into the barrier tissue, but also further transport into circulatory blood. The main routes by which toxicants are absorbed are the gastrointestinal tract, lungs, and skin. However, in toxicologic studies, such special routes as intraperitoneal, intramuscular, and subcutaneous injections are also used. And we have also the pulmonary absorption. The lungs are the primary route of deposition and absorption of small airborne particulate, gases, vapors, and the aerosols. And we have percutaneous absorption. Significant dermal absorption resulting in toxicity may occur for some substances, highly toxic, fat-soluble substances such as organophosphorus, insecticides, and organic solvents. For example, significant absorption is likely to occur after exposure to liquid substances. Percutaneous absorption of vapor may be important for solvent with very low vapor pressure and high affinity to water and skin. And the other routes in toxicology testing and other experiments, spatial routes of administration are often used for convenience, although these are rare and usually not relevant in the occupational settings. These routes include intravenous, subcutaneous, intraperitoneal, intramuscular injections. Now, dear friends, let's focus now on distribution. Transport of toxicant by blood and lymph after absorption by any of these portals of entry, toxicants will reach the blood, lymph, or the body fluid. The blood represents the major vehicle for transport of toxicants and their metabolites. The distribution of a substance with the organism is a dynamic process which depends on uptake and elimination rates, as well as the blood flow to the different tissues and their affinities for the substance. Water-soluble, small uncharged molecules, univalent cations, and most anions diffuse easily and will eventually reach a relatively even distribution in the body. The concentration of a toxicant in blood will govern the rate of its distribution into tissues and the organ, or its mobilization from them into the blood. So, now the distribution of toxicants in the organism, for example, binding and storage. The human organism can be divided into the following compartments. Number 1 is internal organs. Number 2 is skin and muscles. Number 3 is adipose tissue. And number 4 is connective tissue and the bones. The well-perfused internal organs generally achieve the highest concentration of toxicants in the shortest time, as well as equilibrium between blood and this compartment. The uptake of toxicants by less perfused tissues is much slower, but retention is higher and duration of stay much longer. That is accumulation actually due to the low perfusion. 3 components of the major importance for the intracellular distribution of toxicants: content of water, lipids, and proteins in the cell of various tissues and organs. The above mentioned order of compartments also follow closely a decreasing water content in the cells. Hydrophilic toxicants will be more rapidly distributed to the body fluids, and cells with high water content, and lipophilic toxicants to the cells with higher lipid contents, that is fatty tissues actually. The organism possesses some barriers which impair penetration of some groups of toxicants, mostly hydrophilic to certain organs and tissues such as the blood-brain barrier, the placental barrier, and the histohematologic barrier. Now, the retention of toxicant in a particular compound is generally temporary and redistribution into other tissues can occur. Retention and accumulation is based on the difference between the rates of absorption and elimination. The duration of the retention in a compartment is expressed by the biological half-life. Accumulation in lipid-rich tissues. The standard man of 70 kg body weight contains about 15% of the body weight in the form of adipose tissue, increasing with obesity to 50%. However, this lipid fraction is not uniformly distributed. The brain, that is CNS, central nervous system, is a lipid-rich organ, and peripheral nerves are wrapped with a lipid-rich myelin sheath and Schwann cells. All these tissues offer possibilities for the accumulation of lipophilic toxicants. Numerous non-electrolytes and non-polar toxicants with a suitable Nernst partition coefficient will be distributed to this compartment, as well as numerous organic solvents, alcohols, aldehydes, ketones, etc., chlorinated hydrocarbons included, organochlorine, insects such as DDT, some inert gases, for example, radon. Friends, let's now focus on retention in the reticuloendothelial system. In each tissue and organ, a certain percentage of cells is specialized for phagocytotic activity, engulfing microorganisms, particles, colloidal particles, and so on. This system is called reticuloendothelial system, or simply RES, comprising fixed cells as well as moving cells, what we call as phagocytes. These cells are present in a non-active form. An increase of the above mentioned microbes and particles which activate the cells up to a saturation point, toxicant in the form of colloids will be captured by the RES of organs and tissues. Friends, let's now focus on accumulation in bones. About 60 elements can be identified as osteotrophic elements or bone seekers, what we call. The skeleton of a standard man accounts for 10 to 15% of the total body weight, representing a large potential storage depot for the osteotrophic toxicant. Metabolic activity of the bones of the skeleton can be divided into two categories. Number first is active metabolic bone, in which processes of reabsorption and new bone formation or remodeling of existing bone are very extensive. And number second is stable bone with a low rate of remodeling or growth. And incorporation of toxicant into the bone occurs in the two ways. Number first is for ions, and ion exchange occurs with physiologically present calcium cations or anions, for example phosphate or hydroxyl. And number second is for toxicants forming colloid particles, absorption on the mineral surface occurs. Now let us discuss accumulation in the hair and nails. The hair and the nail contain keratin with sulfhydryl group able to chelate metallic cations such as mercury and lead. Distribution of toxicant inside the cells. Distribution to and away from the target. During the distribution phase, toxicant enters the extracellular space and may penetrate into the cell. Lipid-soluble compounds move readily into the cells by diffusion. Highly ionized and hydrophilic toxicants, for example aminoglycides, are largely restricted to the extracellular space unless specialized membranes carrier systems are available to transport them. Now friends, let's discuss biotransformation, elimination and excretion of toxicants. When we talk of biotransformation, it's a process which leads to metabolic conversion of foreign compounds, for example xenobiotics, in the body. The process is often referred to as metabolism of xenobiotics. As a general rule, metabolism converts lipid-soluble xenobiotics to large water-soluble metabolites that can be effectively excreted. The liver is the main site of biotransformation. All xenobiotics taken from the intestine are transported to the liver by a single blood vessel, vena porta. If taken up in a small quantities, a foreign substance may be completely metabolized in the liver before reaching the general circulation and other organs. First pass effective metabolites of xenobiotics can be excreted by the kidney or via bile. They can also be exhaled via the lungs or bound to endogenous molecules in the body. And metabolism is a word often used interchangeably with biotransformation. It denotes chemical breakdown or synthesis reactions catalyzed by enzymes in the body, nutrients from food, endogenous compounds, and xenobiotics are all metabolized in the body. Metabolic activation means that a less reactive compound is converted to a more reactive molecule. This usually occurs during phase 1 reaction. Metabolic inactivation means that an active or toxic molecule is converted to a less active metabolite. This usually occurs during the phase 2 reaction. In certain cases, an inactivated metabolite might be reactivated, for example, by enzymatic cleavage. And phase 1 reaction refers to the first step in xenobiotic metabolism. It usually means that the compound is oxidized. Oxidation usually makes up compound and more water soluble and facilitates further reaction. Cytochrome P450 enzymes are a group of enzymes that preferentially oxidize xenobiotics in phase 1 reactions. And phase 2 reaction refers to the second step in xenobiotic metabolism. It usually means that the oxidizing compound is conjugated with an endogenous molecule. Friends, let's now focus on elimination or excretory portion. Elimination is disappearance of a substance in the body. Elimination may involve excretion from the body or transformation to the substances not captured by a specific method of measurement. During retention in a cell of various tissues and organs, toxicant can expose to the enzyme which can biotransform, metabolize them, producing metabolites. There are many pathways for the elimination of toxicants and metabolites: by exhaled or air via the lungs, by urine, via the kidney, by bile, via the GIT, by sweat, by skin, by saliva, via the mouth mucosa, by milk, via the mammary glands, or by hair and nails via normal growth and cell turnover. Many factors can influence the elimination rate of toxicants and their metabolites from the body. For example, we have the physiochemical properties of toxicants, level of exposure and time of post-exposure elimination, portal of entry, etc. Friends, let's now focus on the renal excretion. The kidney is an organ specialized in the excretion of numerous water-soluble toxicants and metabolites, maintaining homeostasis of the organism. Each kidney possesses about 1 million nephron able to perform excretion. Renal excretion represents a very complex event encompassing 3 different mechanisms. Number 1 is glomerular filtration by the Bowman's capsule. Number 2 is active transport in the proximal tubule, and number 3 is passive transport in the distal tubule. Friends, let's now focus on the elimination of toxicant from the body can be increased by, number 1 is mechanical transcoagulation via gastric lavage, blood transfusion or dialysis. Number 2 is creating psychological conditions which mobilize toxicant via diet, change of hormonal balance, improving renal function by application of the diuretics. And number 3 is administration of complexing agents, for example citrates, oxalates, silicates, phosphates or chelating agents. Or this method is indicated only in person on strict medical control. Friends, let's now focus on what is toxication. Many compounds show direct toxic action to biological systems, for example nicotine. But some compounds must be transferred into the compounds prior to toxicity. Biotransformation to the harmful products is called toxication or metabolic activation. The increased toxic activity of metabolic activation may be due to the conversion of parent compound into the electrophiles, free radicals, nucleophiles, redox active reactants. Friends, let's now focus on ultimate toxicant is the chemical species that reacts with the endogenous target molecule or critically alter the biological environment, initiating structural and functional alteration that results in toxicity. Some of the different types of toxicant and their source are given in the reaction of the ultimate toxicant with the target molecule. The reaction of the ultimate toxicant with the target molecules are as follows. Number first, we must consider what makes a target molecule a target. Number second, what chemical reactions occurs at the target molecule. Number third, the effects of the reaction of the ultimate toxicant with the target molecule. And secondly, we will also consider the possibility of the toxicant to change the microenvironment around critical endogenous organelle biomolecules without necessarily directly integrated with the target molecule. Number first is attributes of a target molecule must possess appropriate reactivity or citric configuration for a non-covalent or covalent interactions and must be exposed to a sufficient concentration of the toxicant. Number second is types of reaction that occurs between toxicant and the target molecules. And non-covalent binding, usually reversible hydrogen bonding, van der Waals force, etc., and typically involves membrane receptors, intracellular receptors, ion channels, and some enzymes. Also, intercalation into double helix of DNA involving non-covalent binding interactions. And number third is effects of toxicant on the target molecule. Number first is dysfunction. Toxicants can disrupt the normal functions of the receptors, ion channels, and even enzymes by binding to the endogenous molecules. For example, protein conformations are disrupted when a reactive species bind to them in vivo. Toxicant can be interfere with the template function of DNA by binding to the nucleotides. Number second is destruction. Toxicant can cross-link or fragment target molecules. An example of fragmentation is a free radical induced lipid peroxidation mechanism, and which breaks down phospholipid cell membranes into reactive aldehyde species capable of further reactivity at sites further down. Number third is neoantigen formation. In some individuals, the covalently bound xenobiotes to a protein carrier the adducts as a hepatin, evoking as immune response from the immune system. And number third step is cellular dysfunction and resultant toxicants. The reaction of toxicant with the target molecule may result in impaired cellular function. Normally, each cell has a defined programs. Programs determine the destiny of cells, that is whether they undergo division, differentiation or apoptosis. Programs control the activity of differentiated cells, determining whether they secrete more or less substance, whether they contract or relax, and whether they transport and metabolize nutrients at higher rates or lower rates. Chemical factors that cause cellular dysfunction. Number first is chemical that cause DNA adducts can lead to DNA mutations which can activate cells death pathways. Number second is chemicals that can cause protein adducts can lead to protein dysfunction which can activate cell death. Pathways protein adducts can also lead to autoimmunity. If protein adducts activate oncogenes or inactivate tumor suppressors, it can lead to uncontrolled cell proliferation and cancers. Number third is chemicals that cause oxidative stress can oxidize DNA or proteins, leading to DNA mutations or protein dysfunction, and all of the above. That is, a good example is of benzene. Number fourth is chemicals that specially interact with protein targets. Friends, let's now focus on necrosis. Necrosis refers to the series of changes that occurs following cell death when the cell is converted to debris, which is typically removed by the inflammatory response. There are two types can be distinguished. One is oncotic necrosis and second is apoptic necrosis. Friends, let's now focus on mechanism of necrosis. Cells must synthesize endogenous molecules, assemblies, micromolecules, complexes, membranes and cell organelles, maintain intracellular environment and produce energy for operation. Agents that disrupt these functions, especially energy producing function of the mitochondria and the protein synthesis will cause cell death. Three primary metabolic disorders of endangering cell survival are: number first, ATP deflection; number second is sustained rise in intracellular calcium 2 positive, and number third is overproduction of ROS RNS. And the apoptosis. Apoptosis is derived from the Greek word apo, means away from, and tosis means to fall. The term falling away from is derived from the fact that during this type of prelethal change, the cells shrink and undergo marked bubblings at the periphery. The bulbs then detach and float away. Apoptosis occurs in a variety of cell types following the various types of toxic injury. The general features of apoptosis are active forms of cell death enabling individual cells to commit suicide, cascade dependent, that is dying cells shrink and condense and then fragment, releasing small membrane bound apoptosis bodies which are phagocytosed by immune cells, and intracellular constituents are not released when they might have deleterious effects on neighboring cells. And when we talk of mechanism of apoptosis or programmed cell death is tightly controlled, organized process whereby individual cells break into small fragments that are phagocytosed by adjacent cells or macrophages without producing an inflammatory response. Sustained elevation in the intracellular calcium 2 positive is harmful because it can result in: number first, depletion of energy reserves by inhibiting ATPs used in the oxidative phosphorylation. Number second is dysfunction of microfilament, and number third is activation of hydrolytic enzymes, and the last is generation of reactive oxygen and nitrogen species, for example ROS and RNS. Friends, let's now focus on phenotypes of apoptosis, and we have overall shrinkage in volume of the cell and the nucleus, loss of adhesion to the neighboring cells, formation of blobs on the cell surface, DNA fragmentation, and rapid engulfing of the dying cells by phagocytosis. And friends, when we talk of factors that induce apoptosis, we talk of internal stimuli, external stimuli, and number third, we talk of signal transducing pathways leading to the apoptosis are two major pathways. First is extrinsic pathway and second is intrinsic pathway. And intrinsic apoptosis is mitochondria dependent and is induced by DNA damage, binding of nuclear receptors by the heat, radiation, nutrient deprivation. Friends, let us now focus on repair or disrepair. Repair or disrepair, many toxins after macromolecules which, if not repaired, cause damage at higher levels in the organism. The organism tries to repair the damaging effects from the toxin on molecular, cellular and tissue levels. Normally, the cellular dysfunction induced by toxin target organ interaction may be corrected by repair mechanisms at the molecular or cellular levels. When these repair mechanisms fail, toxicity occurs. Carcinogenic and mutagenic actions of toxins are due to the failure of repair mechanisms. Some of the mechanisms like protein repair, lipid repair and DNA repair are to be mentioned above. And when we talk of protein repair, protein disulfides, protein sulfonic acids and protein methionine sulfides are reduced by thioredoxin or methionine sulfides reductase. Thioredoxin is regenerated by thioredoxin reductase. Number second is protein glutathione mixed disulfides are reduced by glutaredoxin. Glutaredoxin is regenerated by glutathione reductase. And number second is lipid repair. Phospholipid peroxyl radicals formed from the lipid peroxidation may abstract hydrogens from the alpha tocopherol, which can be regenerated by glutaredoxin, which in turn can be regenerated by glutathione reductase, or what we call as GR. And second is a phospholipase can cleave the fatty acid peroxide, which can be reduced by glutathione peroxidase to give FAOH-GPX is regenerated by glutathione reductase. Quenching of oxidative stress. Detoxification of superoxide anion radical occurs by superoxide dismutase, followed by glutathione peroxidase and catylase. And when we talk of DNA repair is a collection of process by which cell identifies and corrects damage to our DNA molecules that encodes its genome. In the human cells, both normal metabolic activities and environmental factors such as radiation can cause DNA damage, resulting in as many as 1 million individual molecular lesions per cells per day. Many of these lesions cause structural damage to the DNA molecule and can alter or eliminate the cell's ability to transcribe the gene that can affect the DNA encodes. DNA can enter one of the three possible states. Number one is an irreversible state of dormancy known as senescence. Cell suicide is number second, also known as apoptosis or programmed cell death. Number third is unregulated cell division, which can lead to the formation of a tumor that is cancerous. That's all we have in today's program. Hope you understand it. Goodbye.